Prostate cancer is the most common cancer in male population. The treatment is multidisciplinary, and radiotherapy is always part of the treatment. Even 80% of cancer patients will receive at some moment radiotherapy. These patients have a long life expectancy, so quality of life of these patients is very important. Nowadays, we are using so-called advanced modern technique of radiotherapy, intensity modality radiotherapy, VMAT radiotherapy for treat of these patients. Why is important to mention this advanced technique? Because they allowed us to control and precise delivery of X-ray to our patient, and also, which is very important, to spare a surrounding normal tissue. This surrounding normal tissue means the normal organs that are surrounding prostate in this patient, and these are bladder and rectum. These organs can give these patients so-called acute toxicity, which means reaction of this organ during radiotherapy, and acute toxicity is reversible, but unfortunately many years after radiotherapy, and some cases after 20 years, we can late effects of radiotherapy. Planning process of radiotherapy is very complex and start with immobilization, CT imaging, where we can see tumor and surrounding normal tissue. Radiation oncologists do delineation of tumor, but also of this normal tissue, and according to the recommended standard, we know just how every normal organ, which dose can receive and in which volume. After we did in our computer process delineation of this structure, we made the plan and sent this plan to medical physicists. A team of radiotherapy, which includes radiation oncologists, medical physicists, radiation technicians, and of course our nurses, involved in all this process of radiotherapy planning and delivery. Medical physicists are also aware of the constraints of all these organs. They made plan, check plan, send it to radiation oncology, who has to approve the plan, and then this plan is ready to start radiation of our patient. Radiotherapy lasts, radical radiotherapy, usually for 6 weeks, and during that time with our radiation oncologists, our technicians check the field precise of the dose delivery, also according to the protocol. I have to mention that all these I'm mentioning protocol for radiation are also international that we provide for our patients. Toxicity is main interest in this our study, and we did that with collaboration with our colleague from the research. So collaboration from radiation oncologists, doctors, and clinicians, and our colleagues from basic research, hopefully will lead us to make adequate individual plan for every patient. We said we are hoping to do so-called personalized radiotherapy. To find this way to establish toxicity of every patient, we are hoping in the future to find biomarkers who will tell us which patients is more prone to have reaction of radiotherapy than the other patients. This is one of the main goals of our project. But also we can mention that apart from these biomarkers, there are many other clinical and individual factors who are also responsible for radiosensitivity and radiotoxicity. So every individual patient has different reactions to radiation therapy, and we also know for some genes who will tell us which that group of patient is. So the goal of our study, of our cooperation between radiation oncology and our colleagues in basic research is to find possible biomarkers of radiotoxicity and improve quality of life of our patient. Hopefully that results of treatment stay good as they usually were for this group of patients. Because the main goal of modern oncology, of course, is quality of life of our patient.
This is apartment from my experimental oncology in the Institute from Oncology and Radiology of Serbia, Belgrade. My colleague from laboratory from Radiobiology and Experimental Oncology performed a European twinning project, RadExIORSBoost.
Many physical, individual, and clinical factors, in addition to numerous biological factors, may have an influence on the normal tissue radiotoxicity in patients with prostate cancer. In the field of radiation biology and radiation oncology, there is a need to identify novel biomarkers associated with individual normal tissue radiosensitivity and to provide the ability to predict the risk for developing radiation-induced adverse effects for each patient for personalized treatment approaches. To address this research challenge, team from the Institute for Oncology and Radiology of Serbia established a partnership with 4 top-class leading European research institutions: Medical Faculty Mannheim, Heidelberg University from Germany, University of Leicester from United Kingdom, Medical University of Vienna from Austria, and Institute of Oncology Ljubljana from Slovenia. The main goal of our project is to create a multidisciplinary scientific network to address the research challenges in the field of radiobiology and radiotherapy, which will help our institute to further develop its scientific strategy, strengthen its position as a leading regional cancer research institution, and to establish itself at the European level as an excellent research partner for frontier research in molecular radiation biology through increasing its scientific excellence, visibility, and innovation capacity, and the adoption of state-of-the-art techniques and methodologies.
Specific Objective 1 is to develop and implement institutional scientific strategy on radiobiology to significantly increase Institute's research excellence in this field through the transfer of new knowledge from top-class leading partner institutions from Germany, United Kingdom, Austria, and Slovenia. Specific Objective 2 is to examine and further develop predictors of patients' individual normal tissue radiosensitivity by combining gene and microRNA expression in patients' derived peripheral blood mononuclear cells using RNA sequencing, radiation-induced lymphocyte apoptosis, and radiation-induced DNA damage levels and DNA repair activity by COMET assay and gamma H2AX assay, and to design a high-quality clinical study which will lead to personal radiation treatment approach. Specific Objective 3 is to raise the research capacity of researchers from our institution and partner institutions by realization of a comprehensive training program. Specific Objective 4 is to strengthen administrative and research management skills of our institution staff and to upgrade the administrative unit for international cooperation and projects at our institution. Under Specific Objective 5, we aim to manage projects successfully. Specific Objective 6 is to build sustainable communication channels and dissemination of the project results among the scientists of relevant research areas, project stakeholders, and public.
The work plan of our project consists of 6 interrelated work packages. Work Package 1 is development of scientific strategy and increasing research excellence. The main activity within Work Package 1 will be development and implementation of the Institute's scientific strategy on radiobiology to significantly increase Institute research excellence in the area of radiobiology at national and European level through the transfer of new knowledge via 6 visits of distinguished experts.
One approach for assessing radiosensitivity is not sufficient and one test is not specific enough. Therefore, we propose at least 3 potential tests to predict individual radiosensitivity by transcriptome and myrinome analysis of patients' peripheral blood mononuclear cells, and assays for measuring lymphocyte radiosensitivity, radiation-induced apoptosis of CD4 and CD8 positive T lymphocytes, single-cell alkyl comet assay for measurement of radiation-induced DNA damage levels and DNA repair activity, as well as gamma H2AX flow cytometric assay.
Bioinformatics analysis and radiation-induced transcriptome and myrinome changes and pathway analysis will be done to identify genes, microRNA, and biological pathways underlying radiotoxicity. RadExIORSBoost team will test deep learning models from project results on individual clinical parameters and radiosensitivity of lymphocytes to make a predictive model for radiotoxicity in patients with prostate cancer. Based on the results from this work package, we will design a clinical study for validation of biomarkers and models predicting radiosensitivity for prostate cancer patients undergoing radiotherapy.
Work Package 3 will be focused on raising the research capacity of researchers from EORS and project partners through 4 short-term visits, 9 workshops and 6 trainings. Work Package 4 will enable improvement of the administrative skills of EORS staff from financial and legal departments through 6 visits and 2 trainings. Work Package 5 will be devoted to project management and coordination to manage projects successfully, and activities within Work Package 6 will enable project dissemination and communication.
Realization of our project activities may enhance our understanding of molecular mechanisms underlying radiation-induced toxicity, may contribute to scientific efforts to identify novel molecular predictors of individual normal tissue radiosensitivity, and add a new knowledge to establish predictive models and a high-quality clinical study for biomarker validation. The obtained result from the approach combining 3 biological parameters could be a significant step forward to establish a personalized radiotherapy approach, reduced normal tissue reactions to radiotherapy, improve radiotherapy outcomes, and long-term enhancement of the patient's quality of life.